So we are now being recorded. So again, for the purposes of letting those who are still coming in come in, I will not go to the presentation mode yet of this presentation. We'll wait until 2:15 and then I can proceed with a presentation mode. Okay, so again, this is the 10th and last video teleconference for our course BIOL 105L YB for this semester. So unless we need another one, this is the last one. Okay, so if we look at the schedule, this is actually video teleconference number 10. So we are ahead now because we're doing it today, Monday, instead of Thursday. And the reason for that is I might not be able to meet you on Thursday because of a prior commitment. So why not do it today? So this is no longer about an experiment or an exercise, but instead this is on a special topic from the textbook that you have in lecture, which is chapter 16 on cancer. So I think it's very important for you to be aware of how cancer works in the molecular and cellular level. Okay, so I will be giving your final requirement siguro at the beginning of next week. Okay, so for this week siguro parang relax muna tayo and we will just have this chapter um, as something to study for this week. Okay, so let's begin first from where we left off this morning because you are also the same class, no, a while ago. We sort of didn't finish yet on the topic on cell signaling. Okay, so we will continue with that. Okay, so what you see here are pictures, no, a series of pictures of the development of the front limbs, no, the limb bud of a vertebrate, probably human being, because you see 5 fingers here. So notice in the beginning, it's just a bud, it's just a knob, okay? But here in the middle, you can see already that certain portions of the bud are spreading out and you can already see the outline of the fingers. But in between the fingers, we have the webs, no? So webbed finger. Eventually, the webs disappear, and you can see here in the last picture that the fingers are completely separated from each other now. So there's actually an event here that happened in the cells, in the webs, that caused them to disappear. In fact, to die. No, so this is programmed death of the cell. And this programmed death of the cell is known as, what's the term?
Apoptosis, sir.
Yes, okay, so apoptosis. Okay, so the study on apoptosis actually started with a model organism in genetics, no? The kaning roundworm, Caenorhabditis elegans, C. elegans. So C. elegans, all adults of these are known to have 959 cells, always. No? So kung ang mga scientists manakop sila C. elegans nga adults, kung ihapon nila ang cell, it's always 959. Okay, so initially, the cells in the eggs or the embryo of C. elegans is 1090. So what happened to the 131 cells? Those actually died during the development so that in the adults, there are only 959 cells. But of course, we only know this if we encounter the abnormality. So scientists encountered adults that have all the 1090 cells. And they found out that the difference between the normal adults with the 959 cells versus those that are mutated adults is actually the mutation of the gene called SED3. Okay, kaning SED3 nga genes. And these SED3 genes actually contain instructions to make enzymes known as caspases. Okay, so this is very important. When we talk about programmed cell death or apoptosis, the enzymes that should come to your mind should be these, the caspases. Okay, pag caspases na gani ang storyahan, apoptosis din na siya about. Okay. So, what are caspases? They are proteases. So look at this. They are proteases that number one, they inactivate focal adhesion kinases. So kabalo naman, you know what focal adhesion is. Diba? It's used by the cell to hold on to its surroundings. And when that is inactivated by the caspases, the cells will detach from their surroundings. So before the cell dies, it will detach first from its surroundings. No, so very sad. It's like your pet dog or pet cat when it's about to die. Pangitaon din mo, ano naman to si kato akong pet. Okay, ano naman si Brownie. Nagtago-tago na day because it's detaching itself from you because it's about to die. So this is sort of similar to what's happening here in the first bullet, no? And then caspases also cleave the lamins, okay, which actually make up the nuclear lamina. And take note, if the lamins are destroyed, the nucleus will also be misshapen. Mawala ang shape sa nucleus. In fact, here it's said the nucleus will shrink in the dying cell. Then the cleavage of cytoskeletal elements, so katong mga actin, microtubules and intermediate filaments are going to be cut into pieces. So the cell sort of collapses. No, the dying cell collapses. And finally, the cell's DNA are going to be shredded, destroyed by endonucleases. So this is quite a sad description of programmed cell death, apoptosis. But the thing is, it's part of life. You know, your fingers, your normal looking fingers would be impossible without apoptosis. So as sad as this may sound, this is actually normal. And for our bodies, it happens all the time. No? Things that need to be to die, ng mga cells die all the time, you know, using programmed cell death. And it doesn't even hurt because it's planned and it's done in a planned manner, as you can see here, by the caspases. Okay, so we see here, let me present this already. Makitaan pa ninyo akong presentation?
Yes, sir.
Right. So we see here apoptosis and necroptosis. So these are two different things. Okay. Apoptosis is the programmed and proper painless cell death. Okay. The cell is destined to really die at a particular point in time. While necroptosis is cell death because of, say, accident. So for example, nabangga ang imong tuhod dahil nabunog. Katong the cells there die, but they did not die because of natural death. They died because of an accident. So that's necroptosis, no? So here in this picture, we can see both apoptosis and necroptosis. In necroptosis, kaning cell nga dako dire, the cell will leak, no? Mabuslot ang iyang cell membrane and the contents of the cell will leak into the extracellular matrix. So this is not so good. It's necroptosis. Okay, so in fact, this can cause other cells beside it to become sick also. Masakit po ang uban cell kay. Imagine na namatay ang cell, dayon ang iyang cytoplasm or iyang dugo was spread to the other cells. So hugaw, dirty, death. Okay, but here, on the upper right, we can see a cell that's sort of shrinking already. But notice there is no leakage of its cytoplasm. No? So it's like it's shrinking, but it's doing it silently. This cell is undergoing apoptosis. Okay, so that's how you distinguish a cell undergoing apoptosis under the microscope because it shrinks. And then you can see these blebs. So ang tawag ni mga bleb kaning mga mga lobes, no, caused by the shrinking. And then the cell will actually use an enzyme called scramblase. So if you can remember this illustration, this is the distribution of phospholipids, no, and sphingolipids in the cell membrane. The dying cell in apoptosis will use scramblase to observe this. Unsa ning PC? Kinumdum mo unsa ning PC? Phosphatidyl phosphatidyl choline. Okay. So si phosphatidyl choline, sorry, dili si sayop si PS day. So ning PS phosphatidylserine. Okay. So si scramblase will cause phosphatidylserine to actually invert, no, its amount in relative to the inside, no, of the bilayer of the cell membrane. So look at this. Scramblase will cause more phosphatidylserines to be on the outside than on the inside. And this is a signal that will tell white blood cells that are patrolling the area, no, that the cell in apoptosis will tell them to eat me, kill me now. Okay, and the suffering. Okay, so and then the white blood cells will engulf this dying cell, and there is no waste. No, the evidence is all erased. It's as if nothing happened. So that's what apoptosis is. It's planned, it's clean, and it's orderly. Once the cell dies, it's as if nothing happens. It's natural also. Okay. So here's a picture of a white blood cell, no, a macrophage that is already engulfing a dying cell here, no, in the shown here inside the macrophage. So in terms of its molecular biology, how does apoptosis happen? So here we see two paths. One leads to necroptosis. Okay, so start sa necroptosis. In necroptosis, this cell is injured because of an accident. Let's say nabunog. Okay, so a tumor necrosis factor or TNF will be released and will be detected by the tumor necrosis factor receptor, and this will cause a cascade of signal transduction that will lead to the activation of RIPIK, this protein, no, the phosphorylation, which will lead to the activation of MLKL protein, which will form a pore on the cytoplasm. So naay pore, buslot. So because there is already a pore, this will cause the cytoplasm of this cell to leak out. So that's necroptosis. In apoptosis, the same ligand, tumor necrosis factor, will stick to the receptor, and then RIPIK will also be activated. But here it goes a different path. Okay, it actually activates the initiator caspases, okay, which themselves activate the executioner caspases. And when the executioner caspases are activated, they cause the apoptosis of the cell, okay, the planned cell death. Okay, so this is actually an extrinsic pathway because the signal to die comes from the outside. But there is also an intrinsic pathway. Okay, and this intrinsic pathway, if there is cellular damage, there will be signals that will cause the mitochondrion of the cell to leak. No, ang mitochondrion niya mag-leak to release cytochrome C. Okay, if you can remember cytochrome C, di ba, katong sa cellular respiration, sa mitochondria siya makitaan. So mag-leak, mugawas ang cytochrome C. The cytochrome C will cause a signal transduction that will ultimately activate executioner caspases, and then apoptosis will happen. So this one is an intrinsic pathway. So we have an extrinsic and an intrinsic pathway for apoptosis. Okay, so now that we are done with cell signaling, let's go to our main topic this afternoon, and that's cancer. Okay. So cancer is actually a genetic, genetic disease. Okay, when I say genetic disease, it doesn't strictly mean that it can be passed from parent to offspring, not that way. But when I say genetic, it has something to do with the DNA of the cell, the chromosomes of the cell. As you can see in this karyogram of a cancer cell of a human being, the chromosomes are all in um, weird numbers. No, we have a chromosome here, chromosome number 16. Now instead of being a pair, it is now 5, chromosome number 16. And we can see here in chromosome 6 that one of them is taller than the other. So these are hallmarks of the karyogram of a cancer cell, an abnormal cell. So our study on cell actually are done from the cells that came from Henrietta Lacks. No, that was in 1951 when she died of, if I'm not mistaken, I think it's breast cancer. You can just research on that. So while she was in the hospital, there was no rule yet then that you cannot use the cells of a person without authorization. So if this would have happened today, you would need to ask Henrietta Lacks permission that you will use her cells. But during her time in 1951, before she died, there was no rule like that. There was no law. So the scientists just got some of her cancer cells, okay? And Henrietta Lacks succumbed to her cancer in 1951, but her cells, which we now call as HeLa cells, are still alive. No? In fact, you can still actually buy HeLa cells if you want to study cancer cells, for example. No? So this is a testament that cancer cells will really continue living if you just provide them with nutrients. No? In that sense, they are immortal. So that's how cancer cells are. Okay, so these are her cells we call as HeLa cells. Okay, so when we talk about cancer, of course, we talk about multicellular organisms. No, there is no known cancer for unicellular organisms. Okay, so here's a question: is unicellularity a lowly condition? Is it really a bad condition, a sad condition? So we have here a paramecium. Um, Maybe it's a sad condition because if you're unicellular, you can be easily eaten by other predators like this amoeba eating this paramecium. But take note, when the paramecium divides itself, say in binary fission, it's actually cloning itself. Now it can clone itself. And as long as the clone is alive and continues to divide, the paramecium is good as immortal. It can continue dividing again and again and again. So it will always have a clone of itself. So that's why I asked this question, is unicellularity really bad? Okay, kung kamo pangutanon, I'll ask you now, do you think unicellularity is really that lowly? Kamo pangutanon, would you rather be multicellular or unicellular?
Sir, mag-unicellular na lang ko, sir.
Because you can always divide and then the other cell is a clone of yours, meaning to say, ikaw gihapon to. Diba? Just make sure you will not get eaten by predators. Because if you get eaten, then you die. No. So as long as you can hide and continue dividing, you're good. Okay. So is multicellularity really a better solution to survival? Diba? So we have the sponges. We've learned that kaning sponges, if you destroy the sponge, the cells can come back together. Okay? And then we have a more complicated multicellular organism here, like mammals. And of course, humans are the most complex. not so far. So that's what we consider ourselves. Uh, Complex because maybe we have evolved to be able to think, no, and to reason, and then to make tools and to make technology that has caused the world right now to progress. But is multicellularity really a better solution? Steve Jobs died because of cancer, di ba? Pancreatic cancer. And cancer, as I mentioned, only happens in multicellular organisms. So, would you rather be a multicellular organism that has the capability to be immortal as long as you don't get eaten? Or would you be a multicellular organism? You can think, you can make tools, but you are prone to cancer, so you could still die out of cancer. So that's sort of the questions that I have here. So the price of multicellularity is because there are trillions of cells in our bodies. It's very difficult to keep track of all of those cells. By that, I mean that there could be some cells that are already doing um, abnormal stuff and you don't notice them, di ba? Come on, do you keep track of your cells every day? Ah, ikaw normal, ikaw normal, ikaw normal, ikaw normal. All of my cells are normal. We cannot say that because at an obscure part of our body, maybe in our large intestine, there is a cell there that is starting to become abnormal. No, and we don't even notice it. And we only notice it later on when a tumor has formed. So that's the price of multicellularity. It's difficult to keep track of all of your cells, and some of them can become cancer cells. If you can remember Rizal's saying, di ba? Adto ta sa kay Jose Rizal, okay? Ang mga kriminal daw ay cancer ng lipunan. See, Rizal was a doctor. So he's a biologist. So he knows about this, no? The good body is sabotaged by one bad cell. No, ang criminal ay cancer ng lipunan. So, can relate this to his saying. And the word cancer, nganong cancer man ang tawag, sir? Unsa day una, ang cancer nga zodiac or ang cancer na cancer? It's called cancer because as observed under the microscope, the cells tend to spread. And the spreading looks like the limbs of a crab. No? So, maoy lang gi-imagine. So, that's why they call it cancer. Okay? But the original cancer really is the crab. No? And then we just call the situation here cancer because it looks like as if it's a crab under the microscope spreading. And then, not all multicellular organisms are hopeless against cancer. We have found out that this naked mole rat, which has a life expectancy of 32 years, is resistant to cancer. Now, this organism, however ugly it looks like, is actually cancer-free for throughout its life. No, it's almost impossible for this organism to have cancer. Now, you compare that to humans na at ripe age of say 60, most likely you have some tumors already growing in your body. In fact, kana pa lang alom nimo, no, your moles, and if you have moles nga kanang nitubo, those are actually tumors. Those are cells that have run abnormal in their cell division. So mo nang natin kay mole, 'di ba? Not this mole. Incidentally, this is also known as a mole, the naked mole rat, but it's resistant to cancer. Okay, that's what I'm saying. Okay, so what are the properties of cancer cells? Okay, they arise from normal somatic cells. Somatic. So not from sperm or egg cells, but from somatic cells. They defy regulatory influences like cell division. So di sila magpatuo or attachment to the extracellular matrix. Kung ingon mo sila stay, they will not stay. When cultured in vitro, they will continue growing or dividing even when the growth factors have been depleted or even when they're already overcrowded. They divide indefinitely. They seem to be immortal. and they have excess enzymes known as telomerase. No, I hope no makarelate na mo unsay meaning aning telomerase karon. Okay? Um, they genetically are unstable, okay, and they have aberrant chromosomes. So kato kong gipakita ang karyogram a while ago, they have bad conditions of aneuploidy, and they are connected to the topic that we talked about at the beginning of this lecture. They are protected from natural cell death, and they depend on glycolysis instead of cellular respiration for a majority of their ATP production. So that's a thing to remember, no, about cancer cells. Ganahan sila og glycolysis instead of cellular respiration. So here's a general view of how cancer cells are. Okay, so here on the left we have normal cells, epithelial cells, say lining the intestine siguro. Okay, and you can see here in yellow the basal lamina, no, the extracellular matrix. Now, when a mutation in one cell causes the cell to divide uncontrollably, look what happens here in the middle. No, it can sort of block the lumen of the intestine, but sometimes it's benign, meaning to say, although the cell division is uncontrolled, it stays in place. No, the cells stay in place, and this can be removed through an operation. So, pwede lang ka-operahan, i-remove ni nga tumor, okay ka na. But then, the mutation can actually lead to something worse, wherein the cells gain the ability to produce excess amounts of unsa tong enzyme ang mag-digest sa basal lamina?
Itong cancer, matrix metalloprotease.
Correct. Okay. So pwede nga kana excess production of that, and the tumor cell will have the capability to go into the other places of the body, no? Spread into the other places of the body by eating through the basal lamina and the ECM. So mao dining malignant dayon. So maski operahan ni siya nga tawo, kuhaon ni iyang tumor, we could not be sure that in the other parts of the body, the tumor cells have actually established themselves also. Here's a micrograph. So kani picture gyud ni siya, no? So kani on the left is a normal epithelium. You can see the basal lamina here. Okay? Then here in the middle we can see tumor. No, but this could be benign. And how do you know they're benign? Because they are not growing past the basal lamina. So tanawa, they are still on the basal lamina. But here in the right, notice how the cells go through the basal lamina. So this is now carcinoma, as you can see here. Cancer na ni siya kanina sa right. So as long as wala pa siya ability nga mo go through the basal lamina, okay pa, maoperahan pa, it's still a tumor and it's still benign. So here, this illustration shows how they break from one place. You can see here, break through the basal lamina and then migrate. They can move by themselves. They can enter the blood vessel and spread around your body in the different parts of your body through your blood vessel. And wherever they decide to go, they will get out of the blood vessel, they will stay in that portion of your body and they form another tumor. So, mo na mag-spread na din ang tumor, daghan kaayo. So, mo nang ginaingon guys nga, ang una ni mga diagnosis ka na kay bone cancer, pero ang gimong gikamatchan, for example, is liver cancer. So, pwede ana, or the other way around. Ang una nga diagnosis, you have liver cancer, but you died of bone cancer. Because the cancer cells have spread, not just in the liver, but also in your bone marrow, for example. So here's an illustration of normal cells grown in Petri dish, no, sa Petri dish. So, ang normal cell, kung napuno na ang Petri dish, muhunong na sila, they will stop growing. Okay? But if cells are mutated, no, their genes are mutated and they become cancer cells, they will actually divide continuously to the point that they will be on top of each other. So notice ang normal cells is just one sheet, but cancer cells can grow on top of each other and wala sila ipaki kung puno na imong petri dish, they will overflow and they will continue growing as long as you provide them with food. So that's a characteristic of cancer cells. Another characteristic is they prefer glycolysis. So kaning nasa left is a normal cell, no? Remember, we can get more energy from oxidative phosphorylation or cellular respiration than from glycolysis. So in normal cells, they prefer oxidative phosphorylation. So notice this one. But in cancer cells, the one here on the right, they prefer lactic acid fermentation or glycolysis. So that's why maybe you've heard about people who have been diagnosed with cancer and bawal sa ilang mukaon og sugar because sugar is actually the one feeding this glycolysis process, di ba? And cancer cells prefer that. So, mao na kung diagnosed with cancer ang isa ka tawo, bawal gyud mag-inom og gatas, kay gatas also contains sugar or anything na sugary. No, bawal sa ilang diet. And here we can see a picture of cancer cells. They grow to the point nga ang kaning nastunga namatay na lang. Okay? Mao nang cancer cells guys, ang tumor usahay mo sakit siya, ngano mo sakit man kay ang cells nga nasa tunga are already dead. Okay, as you can see here no, na liquefy na, namatay na, nagnana na. And the reason for that is because this environment is anoxic. Kung sa may meaning sa anoxic, there is no oxygen needed for the cancer cells to grow. No, because they grow, they get energy through glycolysis. Okay, so the causes of cancer could be through chemicals, no, carcinogenic chemicals. Like when you handle chemicals in your organic chem, like benzene, no, kadto carcinogen yun ang benzene. It could be radiation, like UV. So mao nang compare mahilig kag painit, UV can actually cause mutations no in your DNA in your cells. And we do not know the mutation can lead to tumor formation. Or viruses. Viruses may also inject or incorporate their genome into your own genes, and they can cause the host cell to become a cancer cell. So a good example for this is in the women, katong human papilloma virus, HPV. So that's why women need to be vaccinated against HPV because HPV can actually cause cancer no, of the uterus, no, when they invade the cells there, I mean, tumor viruses. Of course, diet, no, is very an important cause of cancer. As I mentioned a while ago, if you already have cancer or tumors and you love eating sugar, you are just giving them what they need. And chronic inflammation. No, for example, nakay hubag. Dahil wala ni magipatanaw sa doktor ni samot. No, ang hubag dugay na kay nga hubag. Imong ipon lugar naghubag. Dahil mahadlo ka mo adto sa dentist. Nag 1 year na lang tong hubag sa imong ipon, that can actually stimulate the cells to become cancer cells. And of course, this is something that we cannot run from. It could be genetic predisposition, no? Kaliwat, cancer. Cancer is monoclonal. It can start with a normal cell here. So kaning gray normal cell. The normal cell could say have a mutation. So marining blue. And then this mutated cell will of course undergo cell division. Okay, cell division. And then one of its descendants underwent another mutation. So kaning yellow-orange. And then this yellow-orange cell will divide again in mitosis. and one of its descendants may undergo another mutation. So magpatong-patong ang mutation. Okay? And then you have these green cells, no, and you have these purple cells, and then eventually a descendant of which becomes a cell that is a cancer cell. No, not only a tumor cell, but a cancer cell able to spread to the other parts of the body. So, ana ang cancer, guys, it's not just one mutation. It is a combination of many, many mutations, no, in a particular cell that has caused it to become a cancer cell. So it's monoclonal, meaning to say its origin is from a one cell. Gikan siya sa isa ka cell. Here's another illustration of that. No? So, gikan sa isa ka cell, the cell mutates, no? Nagalahi ang color, meaning to say lahi nga mutation, hantod ni abot aning last nga mutation which has the capability to eat through the basement membrane. Okay. That's why from a normal karyogram, it becomes so weird looking like this because this is accumulated mutations in the cancer cell. So let's look at the genes that are involved in cancer. So kani important. No? First, we have the tumor suppressor genes. What are tumor suppressor genes? Normally, these genes help your cell be under control when they divide. So, kung mao ni sila nga genes nga moingon nga, oh, don't divide yet. You don't need to divide. Okay, that's what tumor suppressor genes do. Okay? But when they are mutated, they lose their ability to control cell division. And therefore, they can contribute, no, pag nang-mutate sila, they can be implicated to cause cancer. So here's an example. So take note that genes come in pairs, no, one from the mother, one from the father. So notice here, this cell divides, the other cell is normal, while the other cell has a mutation on one allele of the tumor suppressor gene. This cell is still normal. Why? Because the other normal allele for the tumor suppressor gene can still do its function. So, okay pa, normal. The trouble happens when both of the tumor suppressor genes are mutated. That's when the cells lose control in their cell division and can become tumor cells. Okay, that's the tumor suppressor genes. The other gene type implicated in cancer are the proto-oncogenes. What are proto-oncogenes? These genes, they function in, say, in cell division, gihapon, control in cell division. Okay? But proto-oncogenes, once they are mutated, or once they are duplicated, or once they lose their regulation, okay, they will continue doing their purpose in cell division that will enhance cell division. So, how do we differentiate this? Ang tumor suppressor genes, originally, they were to control cell division. Ang proto-oncogene, ang iyang function is to help in cell division. So, kung ang tumor suppressor gene na daot, wala mag-control sa cell division, cancer. Kung ang proto-oncogene na daot, mabuang siya, it will always tell the cell to divide. Ang resulta, cancer gihapon. And here, you only need one copy of the proto-oncogene to mutate to have cancer. Okay? So, adequate na ang isa lang ka-allele ang mag-mutate sa proto-oncogene to form cancer. So, these are the two main types of genes that are involved in cancer formation. So let's start with an example of the tumor suppressor gene, the RB gene. Okay. Take note, genes are named with capital letters na italicized. Okay? So kung makakita gani mo ani RB, that's the name of a gene. Capital letters italicized. And genes are usually named after the disease that happens when they are non-functional. So, kaning RB gene is named after retinoblastoma. Retinoblastoma happens when the RB gene mutates. Okay? So RB gene ang tawag. Now, retinoblastoma in the general public is very rare. Okay, this tumor in the retina, very rare in the general public. But in certain families, there is 90% incidence in a particular family. So what does that tell us of retinoblastoma? Is it hereditary or not? Anyone? The general population, rare daw, but in certain families, very common. So does this tell us that retinoblastoma is hereditary? Yes or no?
Hereditary siya, sir.
Okay, it's hereditary because in certain families, it's there. No, it's present in certain families. So what can you say about a family? Diba? The parents pass their genes to their offspring. So in certain families, it's high in incidence. So it must be inherited. So correct. So in the normal population, this is what happens. This is a tumor suppressor gene. In the normal population, both of the RB gene now start as normal. And the mutation of one of them happens, but this is mutation is actually quite rare, no, in rare in case or in incidence. So in the normal population, they start as normal, both RB genes. So even if nag-mutate ang isa, but the other RB gene is still okay, okay pa. Wala pa retinoblastoma. The retinoblastoma will only appear when both of the genes are mutated. And this chance na both will become mutated from both normal is very rare. And that's the reason why in the general public, retinoblastoma is rare. But in certain families, kunwari si mama o si papa nag-asawa, no? Then both of them actually already have a mutated RB gene, then there is a very high possibility that they can pass the mutated RB gene to their offspring. And therefore, mas high ang possibility, 90%, that their offspring will have retinoblastoma. So, it's inherited. Okay? So, this is the case of retinoblastoma. So, unsa man ang function aning RB gene? Okay. So, si RB gene, as we see here, is the one that makes the protein RB. So, morning PRB, this is a protein. The protein RB actually prevents the transcription factor, the green one here, from transcribing this DNA, which can produce mRNA to produce a protein, which can promote the passage from G1 phase to S phase. So, ang trabaho ni RB gene is to make the RB protein which prevents this from happening. Okay? By default, it prevents it from happening. Only when it's phosphorylated will it allow the production of this protein which can cause G1 cells to become S cells. Okay? So, pag nag-S phase na ganina, diretso na siya cell division dayon, di ba? Mitosis na dayon na. G2 and mitosis. So, if your RB gene is mutated, therefore, the protein RB that it produces will not be able to function. So, instead of by default stopping this transcription of this mRNA, by default, ang faulting RB gene will produce a protein RB that doesn't do anything at all. And the transcription of this DNA continues on and on and on and on without stopping because wala man imong protein RB, non-functional man. So you always produce this protein that will stimulate the cells to undergo S phase, this no matter what. So, kung sa may implication na, this cell will continue dividing because it's constantly being told to proceed to S phase. No? So, mao na si RB gene, retinoblastoma gene. Okay. And then we have this P53 TP53 gene, another tumor suppressor gene. Okay. This gene produces a protein called P53, kaning blue-green. No? This P53 is known as the guardian of the genomes. Okay. Meaning to say it protects your DNA from damages. No, or it helps detects damages so that they can be repaired. In fact, in most of the cancers, no, a large mutation is actually in that TP53 gene, no, as shown in this graph. Unsa may function ng TP53? So here we have cells that are normal. No, when this cell that is normal divides, it can proceed. So, okay, but when there is DNA damage, okay, before the cell can divide, the P53 helps detect the damage so that the damage can be repaired. And when the damage is repaired, that's only when the cell may divide. So stop muna kay na-damage ang DNA. That's what the P53 protein does. No? And if the damage is irreparable, the cell will die. That's because of P53. Now, if there is no P53, like the one shown here, even if there's DNA damage, the cell will still continue dividing. No, and of course, we can expect this to continue dividing, so the damage sort of builds up. And so the cell eventually becomes a tumor cell. So that's why P53 is known as the guardian of the genome, because it helps detect DNA damage prior to cell division. No, so if you can remember this diagram, we discussed this in the previous video teleconference. No, where's the P53? It's here, this one. No? Okay, and it helps here the transcription of P21, which actually sticks with a cyclin-dependent kinase to help it from sticking to the cyclin, so cell cycle is arrested. We have this diagram as an evidence. And then here we can see electron micrographs of cells that have here sa taas both TP53 alleles are okay. So notice, these are actually, by the way, these are chemotherapy drugs. Yung kanang pampatay sa cancer cell, no? Okay. So, if both the TP53 alleles are okay, look what happens. This is untreated. And then, when treated with chemotherapy drugs, the cells die. Okay. And because of apoptosis. Now here in the middle, we have one of the P53 alleles na okay, one is non-functional. And look what happens. Even if the chemotherapy drug is administered, there are still cells that survive. What more if both of the P53 gene or alleles are damaged? Look at this. Chemotherapy does not have an effect at all. So mauna guys, kung level 4 cancer or stage 4 cancer, most likely the cancer cells TP53 alleles, both of them might have been destroyed already. So wala gyud effect ang chemotherapy aside sa maupaw ang patient. So that's what we can say, no? Okay, about chemotherapy, and if you know what it is, okay, and how it works, the prognosis of survival of a patient na stage 4 cancer is really small because of this mutation of the TP53 alleles. This one is the breast cancer gene, so called BRCA, which also helps in the repair of DNA damage. Okay. And then let's talk about oncogenes. So ato na lang ning humano ning oncogene, kaya hapit ng time. So oncogenes only require one of the alleles to mutate before the cell can become a cancer cell or a tumor cell. And there are many genes that are implicated as oncogenes. There are those genes that make the growth factors, no? what if the gene that makes the growth factors is mutated? So the growth factor can stick to the receptor and never detach. So if the growth factor is always stuck here, then the instruction is always to divide. Kay growth factor man siya. Divide, 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 divide. So the gene that instructs for the making of the growth factor proteins are proto-oncogenes. Okay? Also the receptor proteins. What if the receptor protein gene is faulty and it makes a receptor that never lets go of the growth factor? So mura kag kanang nag on bitaw ka sa imong computer dayon dili na ma-off imong computer hantod malubat na lang tungod kay naay virus. It continues on and on and on and on and on. In this example, the cell continues on and on dividing. And so ang resulta tumor, no? And so we have other proteins here. Notice RAS. RAS comes back in the story again, no? Mao na itong topic this morning. If the gene that makes RAS is faulty, no, it becomes an oncogene because RAS works in the pathway. Remember the RAS MAPK pathway for cell division. Okay, if this is faulty, it will cause the cell to continue dividing without any control. And you also have other genes here implicated, no, in this illustration, which I will not detail anymore. And then there are people who have so many mutations in different genes that they have what we call as the mutator phenotype, meaning to say they are really at risk at having cancer. No, gamay lang kaayong exposure to radiation or to the chemical, pwede na dahil sila magka-cancer even without an exposure. So ang tawag nato sa ilang mutator phenotype. Okay. Some strategies in combating cancer, immunotherapy, you can research about this or read about it. Inhibiting the activity of cancer-promoting proteins, targeting the right cells. So, kung chemotherapy, are you really targeting the right cells? Or are you just targeting the hair follicle cells mo na mga lagas ang buhok, pero ang cancer cell buhi gihapon? Inhibiting the formation of blood vessels, no? Because cancer cells depend on blood. So you might want to stop their blood supply. Okay, so these are some strategies in combating cancer cells. I'm done with my lecture on cancer cells. Are there any questions? Interesting kayo siya, no? If we have more time actually, we can really talk a lot about the molecular biology of cancer. Questions about the lesson? Were you still able to see my presentation a while ago?
Yes, sir.
Tama na tong last na ko nga slide katong control
Yes, sir.
strategies. Okay, good. Okay. Ni auto ko ano naman gud siya nawala. Okay, so wala mo questions? If you read your book, there are more details from there. Okay, so this is our last video teleconference. I will just be giving you your last requirement online in eLearn. So just check eLearn early next week. Now I will give your requirement. So I guess if you have classes next sem na ako gyapon ang maestro, we will see each other. Okay, but for now, this is our last video teleconference. So wala na questions?
Po, sir.
Wala na, okay. That's good, no? So read about chapter 16, cancer as well, and cell signaling. If there are no questions, guys, thank you very much for listening. I enjoyed talking. I hope you enjoyed listening. Okay, so you will see the recording of this soon in our course wall. I will just message you. So for now, I hope you'll have a productive remaining semester ahead. Okay, study for your exams. Okay, and thank you very much for joining. You are now all dismissed.
Bye, sir.
Thank you, sir.